Hello everyone, and welcome to the first webinar in our four-part Foodborne Pathogen webinar series. My name is Laura Wilde, and I'm the Senior Program Analyst in Food Safety for the National Environmental Health Association. It is my pleasure to facilitate today's webinar. I am joined by my colleagues Maddie Gustafson, Project Coordinator, and Taryn Laird, Project Public Health Communications Specialist. Please note this webinar is being recorded. If you are not okay with this being recorded, you may disconnect at this time. Today marks the first day of National Food Safety Education Month. NEHA is dedicated to supporting our food safety workforce, and we have several free events and webinars throughout September. Take a look at our website where you'll find a food safety declaration signed by President Long, a message from our Executive Director, Dr. David Dijak, and information on the next 3 webinars in this Foodborne Pathogen webinar series. We are also pleased to highlight food safety heroes by highlighting some of the great work accomplished by you, our food safety professionals. Submit your food safety hero to be recognized for their hard work and dedication to their profession. Additionally, membership does have its benefits. We create and deliver value every day for you. Connect with your peers, network, and get access to valuable resources and educational materials. If you are not already a member of NEHA, check out our website at neha.org to learn more. A little bit of housekeeping before we dive into Salmonella. Attendees will be in listen-only mode, and as a reminder, this webinar is being recorded. We intend to have time for questions at the end of the presentations. So please submit any questions you may have in the Q&A box, and we will do our best to answer as many questions as time permits. I'm pleased to introduce our speaker today. Dr. Donald W. Schaffner is a distinguished professor and extension specialist working in food science at Rutgers University. His research interests include hand washing, cross-contamination, and quantitative microbial risk assessment. He is a fellow of the Institute of Food Technologists and American Academy of Microbiology and International Association for Food Protection. Dr. Schaffner was the president of IAFP in 2013 and 2014. In his spare time, he co-hosts the Food Safety Talk and Risky or Not podcasts. Since the start of the pandemic, he has been interviewed for TV and print media, including New York Times, Washington Post, and Wall Street Journal, on SARS-CoV-2 and the risks of COVID-19. Welcome, Dr. Schaffner. The floor is yours.
Thanks, Laura. It's a pleasure to be here. Let me get started with my screen sharing. Give me a second here. To go into presentation mode. Okay, so so thanks to NEHA for the opportunity to come here today. There's 2 kinds of presentations that I give, sort of standard stock presentations and new presentations. And this one is a new presentation, although you'll see there's a lot of, I wouldn't say recycled content, but there's a lot of content that I'm familiar with just because I'm going to be drawing on some of our own research experiences. And when Laura approached me about doing the webinar, and she said, we'd like you to talk on Salmonella, what should the title be? And you know, one of the things that immediately comes to mind whenever I think about Salmonella is if you really want job security as a food microbiologist, you could do a lot worse than to pick Salmonella. And so hopefully what I'm going to illustrate for you in my talk today is just how interesting this organism is and what do I mean by job security. So yeah, and so I'll also say, again, this is a brand new presentation. So I'm trying it out for the first time on you guys today. Hopefully it will go smoothly. And I'll sound like I know what I'm talking about. So let's get into it. I thought I would open with this obligatory historical perspective. Salmonella has been with us a long time. The organism was first visualized in 1880 by Carl Elberth, and he saw the organism in Peyer's patches and in the spleens of patients that were suffering from typhoid. The first actual discovery of Salmonella was made by a scientist named Theobald Smith. He discovered Salmonella enterica, the variant cholerae suis, in 1885. So we've had more than 100 years of this particular microorganism. In looking up a little bit about Theobald Smith, I discovered he is about 102 years older than I was, so it made it really easy to follow his career. So he's just a few years ahead of me, and plus 100 years. So in 1885, Theobald Smith was working in the veterinary division of USDA under a scientist and administrator named Daniel Elmer Salmon. And that'll come back, obviously, in just a minute. Originally, we thought that Salmonella cholerae suis was the causative agent of a disease called hog cholera. And so Salmon and Smith named the organism hog cholera bacillus. And so, of course, bacillus means rod-shaped microorganism, and so that's a nice logical name. The only problem was it turned out that hog cholera was actually not caused by Salmonella cholerae suis. Hog cholera was actually caused by a virus, but of course our understanding of viruses, especially in the 1880s, was not nearly as advanced as it is today. So, and then about another year later, Joseph Leon Rangeres proposed that the organism be renamed Salmonella in honor of Daniel Elmer Salmon. So again, Smith is the one that discovered it, but because Salmon was his boss, it got named for Salmon. And I was really interested. I knew Theobald Smith had played a role in this, and I'm interested in Smith in particular because the local branch of the American Society for Microbiology that I am a part of is actually called the Theobald Smith Society. And the reason for this is that Smith, a number of years later, in 1915, came to run the Department of Animal Pathology at the Rockefeller Institute for Medical Research, which is in Princeton, New Jersey. And then he stayed there throughout his career, and he retired and became director emeritus in 1929. So there's definitely a Rutgers connection, although the organism was not discovered in New Jersey. There is definitely a Rutgers connection. So now that we've dispensed with the obligatory historical perspective, we will cover the obligatory discussion of the basics of the organism. As I've implied, it's a rod-shaped bacillus. It happens to be gram-negative. It grows over a fairly wide temperature range, not as wide as some organisms. It'll grow all the way down to 55.2 degrees C and all the way up to 46.2 degrees C. For those of you who are more comfortable thinking in degrees Fahrenheit, that means it grows between 41 and 115. It grows over quite a wide pH. It's been documented to grow in foods with pH as low as 3.7. Now, or I shouldn't say foods. It's been shown to grow in laboratory media where you tweak the media and you make everything else optimal. You can actually get it to grow down as low as pH 3.7. I think it's you'd be hard pressed to see much growth in foods at a pH of 3.7. But again, that's what it's defined as in the literature because of some experiments that have shown that. It can grow down to a water activity as low as 0.94. So again, not as low as some organisms, but definitely grows down to those low water activity conditions. It is an incredibly destructive organism. It causes more than 1 million estimated foodborne disease cases every year. This is the estimate from the CDC paper by Scallon et al. We don't actually have 1 million recorded cases. We have a smaller number of recorded cases, but we know that there's underreporting factors. And so we get that estimate of 1 million. But there's uncertainty in that estimate. It could be as big as, or it could be as low as 640,000 and maybe as big as 1.6 million. It is the number 2 cause of foodborne disease in this country, but it is the number 1 bacterial foodborne disease. Of course, norovirus is number 1, but Salmonella is the most prolific in terms of bacterial foodborne pathogens. So what foods does it cause have been associated with Salmonella outbreaks? Well, this is a partial list that or not foods, but what vehicles. This is a partial list. I brainstormed most of this last night from memory with a little bit of searching on Google. I reached out to a few friends of mine via text message today, and they added a few more things to this list for me. So you can see there's a lot of foods here, but there's also interesting things like backyard chickens. CDC has a number of papers about outbreaks from people raising chickens in their backyard. Interestingly, my podcast co-host Ben Chapman recently started a pandemic experiment where they have he lives down in North Carolina, where the zoning laws apparently are not so strict, and he's got a bunch of chickens in his backyard, and so we'll find out if his family gets sick. He's keeping track. Also on that list, you see bioterrorism. I don't know how many of you know about the bioterrorism event that used Salmonella. There was a cult in Oregon, the Sri Rajneesh cult, and were trying to skew a local election, and so they went around and they sprayed Salmonella on salad bars in the restaurant. The Salmonella strain that they used was a high isolate from a hospital, and so the epidemiologists figured that out pretty quickly. But it actually was a potential bioterrorism cause. Going down that list, you can see a whole bunch of different foods. At the top of the list in the middle, you can see food handler. And so obviously, anytime a food handler handles any food, they can cause Salmonella. Let me see if I can make my pen here. There we go. So food handler at the top of the list. So that means it could be basically anything. Another interesting one is Kratom. Kratom is a dietary supplement, and you may have seen some recent cases in the last couple of years linked to Kratom. Marijuana, this was one that one of my friends told me via text message. It's actually been linked to transmission by marijuana. Let's see, what else have we got on the list that's interesting? Well, I think that's it, and there's a lot of other foods. Oh, in addition to, well, I should say frogs are on that list. Let me see what happened to my pointer. Oh, here we go. Yeah, so frogs are on that list. Turtles are on that list. The frogs were actually what we think caused an outbreak that was linked to orange juice. We found frogs in the orange field, in the field where the oranges were being raised, and we found the same Salmonella on the frogs as in the orange juice. And then, of course, if you've been watching the news, you know that, again, I lost my pointer. We know that onions have been in the news, and peaches as well, as being both being linked to Salmonella. So certainly, this is a part of the reason for my answer of why you would have job security. Again, just a wide variety of foods. Raw animal products, meat and poultry, vegetable products, dry foods, and we'll come back and we'll circle around and talk. Shellfish is on the list. We'll circle around and talk about all of these, or many of these, in turn. So now that I've sort of given you that foundation, from here on out, it's going to be all about me. And what do I mean by that? Well, I figure that what I know best is the research that we've done in my lab, or that I've been a part of. And so basically what I'm going to walk you through in the next about half an hour or so is just basically snapshots, tables or figures from research that we've done over the years that illustrate just how interesting and diverse this microorganism is. So with that, I'll move on to the next slide. And this actually, whoops, if I can get my computer to advance. Well, let's see here. Well, give me a second, having a little technical difficulties here. There we go. Nope. There we go. Okay. Oh no. Let's see. I've got all these annotations. Let's see. Well, this is what you get when you try to innovate and fly by the seat of your pants. So you guys are can one of you moderators confirm that you guys are seeing these annotations on the screen when I go to the next slide?
We are seeing them, yes.
All right. So let me annotate. Let me erase. I'm going to erase all these annotations, and I apologize. You know, Zoom is a great platform, except when it's not. All right, so that's that. And now
We
let's go on. All
appreciate the
right.
technical innovation here. It was
Thanks.
good.
Thanks. All right, so let's get down to it, and let's talk a little bit about some of the work, like I said, that we've been doing in my lab. So, or in related labs. So I started at Rutgers in 1989, but I actually did not get into doing Salmonella research until about the mid 2000s, and this was my first foray into this topic. This is a paper by Michelle Daniluk, Linda Harris, and myself. Linda is a faculty member at UC Davis. Michelle was her graduate student, and Michelle is now a professor at the University of Florida. And Linda reached out to me and she said, hey, look, I got this big project on Salmonella in almonds. Would you like to help me with that? Do you think we could do a risk assessment? And I had done a few simpler risk assessments before, but Linda really intrigued me with her idea, and so we began to work on this risk assessment. And of course, Michelle did all the hard work, and Linda and I just provided advice. This paper was published in 2006. Most of these papers were published in the Journal of Food Protection. If anybody would like a copy of any of them, I'd be happy to make them available. Just let me know. So this is table 5 from that manuscript. And basically, let me just walk you through some of the stuff. So one of the things that we wanted to know was what would be the effect of establishing a 5-log reduction for almonds with a certain degree of uncertainty. So 5-log reduction plus or minus 1 log, which again, if you know anything about how almonds are treated and roasted, any process that you give is not a perfect process. There's always some variability in that process. And then we also knew that occasionally with almonds, this is based on some research that Linda did, occasionally with almonds, you have a high contamination event. And so we said, well, okay, what is the effect of that frequency of that high contamination event? So most of the almonds come in, they're contaminated at a low level, but occasionally you'll get a really contaminated batch. And we estimated worst case, based on some data, again, Linda had collected, about 1 in 64 samples would have this high level of contamination. But what you can see here is that frequency of high contamination doesn't really affect the risk all that much, okay? And so if we were to continue eating raw almonds, we would say, without any treatment, we would say there's a fairly high probability of having more than 1 case a year. We even have about a 50%, a little bit less than 50% probability of more than 10 cases a year, and even as much as 20% of having more than 100 cases a year. So this was a clear signal to the almond industry they needed to do something. And so one log reduction that's very popular is a 5-log reduction. And so in our manuscript, we looked at a 5-log reduction. Now, where the almond industry of California, specifically the Almond Board of California, eventually netted out was to require all almonds that are produced in the state to have a 4-log reduction. And so that's a risk management decision, right? And you get similar risk reductions with a 4-log reduction. But again, I think this shows pretty clearly that if you didn't do anything based on our assumptions about baseline levels of Salmonella in almonds, you would continue to have outbreaks. And our analysis shows if you mandate a treatment, those would pretty much go away. Almond Board of California does require a 4-log treatment for all almonds produced in California, and since they've mandated that, we haven't seen any outbreaks. So good news. So that was 2006, although it seems like just the other day. This is a paper that I published with my graduate student Bin Liu in 2007, and here we were looking at the effect of we're going to change foods, okay? And so this is going to be a whirlwind tour where we change from one food to the next. This is an example of where we were looking at Salmonella in sprouts. And in particular, we've done a lot of research with this organism, Enterobacter aerogenes. It's a gram-negative organism, and we sort of treat it in our lab as a biosafety level 1 organism that is less pathogenic than things like Salmonella and E. coli. And we do a lot of experiments to see if the behavior of the organisms is the same. And basically, the idea here is we were wondering, well, what if we started with different levels of Salmonella? Does the level of Salmonella that you start with when you start with contaminated sprouts? Does that make a difference in terms of the level of contamination you end up with in the final batch of sprouts? And the short answer is yes, there actually is a difference, and that difference is because of something called the Jameson effect. The Jameson effect is not the effect that you get from drinking too much Jameson whiskey. It's a different effect, and basically what the Jameson effect says is that if you have multiple microorganisms growing in a food system, they're going to all grow, but as soon as the dominant organism reaches stationary phase, they're going to have basically tied up all of the available food in the system, and so all of the other organisms stop growing. So you can see that here, and I'm going to risk making an annotation, which I'm going to have to erase later. But I want to point out to you that right here, this is a total plate count. When total plate count reaches about 10 to the 8th, you can see that basically shuts off everything else from growing. Now you might see a little bit of a bump in terms of the level of the other organisms, but essentially they stop growing. And if you started at a lower level, you're going to end up at a lower level versus if you started at a higher level. So, and again, that's just a little bit of a glimpse into some of the work that we've done with sprouts. So with that, I'm going to turn that off and go on to the next slide. Oh, okay. So, all right. So we started in almonds. We moved to sprouts. We're going to jump now to poultry. This is Salmonella in poultry. It's a paper by my graduate student, Sylvia Dominguez, we published in 2008. And one of the things that we do in my lab, as you heard in the introduction, we do a lot of risk assessment work. And this is one example of risk assessment work, but also modeling data from the literature. And so what we did here is we collected a whole bunch of papers. People love to study Salmonella in poultry, and you can see that list of papers over there on the right-hand side. At the time that we had published it, the most recent paper was from Tom Oscar in 2002, but this goes all the way back to Alfred and Palumbo, published a paper in 1969. So we basically took all of those data from the literature. They studied a variety of different temperatures. We extracted data from those papers. We plotted the growth rate. We estimated the growth rate in terms of the change in log CFU per hour, and then we plotted that as a function of temperature. And what you can see is this nice typical curve that you see with a variety of microorganisms whenever you look at on a growth rate versus temperature scale. You see a plot that looks something like the one that you see here. So I'm just going to erase a few more things. There we go. So yeah, and so what you can see is that despite despite different laboratories using different strains, different laboratories using different strains, different media, basically you can see, and there's some variability, right? And you can also see the really popular temperatures, right? A lot of people like to study 20 degrees. A lot of people like to study 25 degrees. A lot of people like to study 37 degrees. But what you can see is that although there is some scatter, there's generally quite a bit of agreement in those data points in these studies. And so again, we were able to construct a mathematical model which describes the growth of Salmonella as a function of temperature. Actually, within a few years, I was able to use that to help out a food company, a poultry processing company that had a process deviation. And I was able to show through using our model, which was based on poultry, that in fact their deviation did not result in a significant amount of Salmonella growth. All right, so we're going to switch from poultry now to cut tomatoes. And this is a paper by my graduate student, Wenjing Pan, which was published in 2010. We were studying the growth of Salmonella in cut tomatoes. And again, as mentioned, I do a lot of research in my lab with mathematical modeling. One of the things that we like to do is we like to make our own models. And then we also like to compare our models to the models that other people have developed. And so what you can see here is that our tomato data are shown with the black circles, and we compared the model from a popular modeling program called Combase, which is the dots and dashes, versus the pathogen modeling program, which is the solid dashes, versus our raw poultry model. from the previous paper. And what you can see is that basically our raw poultry model actually does the best job. Com-based underestimates, the PMP underestimates at low temperatures and overestimates at high temperatures. But generally speaking, the raw poultry model was the best one for describing the growth in tomatoes. One interesting thing about this research is that we specifically got outbreak strains, human isolates from the CDC to use in this research. We wanted to make sure we didn't want to just use any Salmonella willy-nilly. So we went through all the paperwork, and believe me, there was a lot of paperwork to get the specific strains of Salmonella that were clinical isolates from humans who ate tomatoes and were part of this outbreak. So that's our research on cut tomatoes. We're gonna change gears once again. We're gonna move from tomatoes. Oops, excuse me. We're gonna move from tomatoes to ceviche. So this is a paper that was published in 2013 with my graduate student Pratik Mathur and myself. Interestingly, I visited another former graduate student. She didn't work with Salmonella, she worked with Bacillus, but I had visited her in Peru, and she took me for ceviche, and it was wonderful, and I didn't get sick, but I know that ceviche can pose a risk of foodborne disease. And interestingly, no one had ever studied, actually, as far as I could tell in the published literature in 2013, no one had ever actually studied the effect of lime juice on inactivation of Salmonella. So, or Vibrio for that matter. And so my graduate student Pratik, we decided he was going to work on this particular project. So he did a whole bunch of research on seafood. He later told me that he comes from one of those states in India that's in the internal part of the country, and he had never seen so much seafood in all of his life. But yet, nevertheless, he worked successfully on this project. I don't have the data on Vibrio because this is a seminar, is a lecture on Salmonella. Lime juice works pretty well for inactivating Vibrio on fish. It does not work nearly so well to inactivate Salmonella. And so we studied a bunch of different things, a bunch of different variables in this experiment. We looked at recovering on two different types of media. We looked on XLT, which stands for xylose lysine tergatol, XLT4, which is a common Salmonella recovery medium, and then we looked at treatment times for treating the fish with lime juice for 30 minutes or 120 minutes. We also looked at recovery on tryptic soy agar, and I think in this one we were using antibiotic resistant Salmonella, so we could recover it on the tryptic soy agar. And then we also looked at treatments of, like I said, 30 minutes and 120 minutes. And then we looked at treatment at room temperature and at refrigeration temperature. And basically, if you look at this is the mean log reduction in the concentration of Salmonella. So what you see is there really isn't too much of a pattern, right? There's not much of a difference between room temperature and refrigeration temperature. Maybe you get more kill with refrigeration temperature, but only on the XLT agar. That's probably because the cells are being stressed by XLT. It's a selective medium, and so it's not good for recovering injured cells. So probably what's going on is that the cells there are injured. You can see room temperature versus refrigeration temperature doesn't make too much difference if you're recovering on tryptic soy agar. And then you can also see there's really not much difference between 30 minutes and 120 minutes. It looks like at room temperature, you get less kill at 120 minutes, and maybe you get more kill under refrigeration. But again, it's all about in the range of about a half a log reduction to a 2 log reduction. So again, lime juice is not ineffective at inactivating Salmonella on fish, but it certainly is no 5 log reduction. So if you like and enjoy ceviche, by all means have at it. But I will probably limit my consumption. All right, so we're gonna switch gears now one more time from fish to where you can find fish, which is in the water. This is some data. This was done in collaboration with Rachel McEgan, who was a graduate student at the University of Florida. And this is, again, a flashback. Remember, that first paper was done with Michelle Daniluk, who was a graduate student at UC Davis. I mentioned Michelle is a faculty member at University of Florida now, and Rachel is one of her graduate students. And so Rachel did this work. They were assisted by my graduate student, Gabriel Mudian, who's another co-author on this paper, and we published this in 2013. What we were looking at to try to figure out here is if you are looking to prevent Salmonella and you test for indicator organisms like E. coli, how good an indicator is E. coli for the presence of Salmonella in waters in Florida in this particular region where we tested them? And these are not actual data, these are regression models, but they're regression models that are based on data. And now, let me just take a minute to walk you through this. It's a little bit complicated. And so this is the probability of enumerating different levels of Salmonella based on MPN as a function of the MPN level for E. coli, right? And so the solid line at the top at about 80%, that's the probability of having more than 3 MPN Salmonella per 100 mil. And what you can see is that line is fairly flat, right? So what that says is that as the level of E. coli increases, it doesn't really tell you very much about low levels of Salmonella in the waters that we tested. What you can see as you move down that curve to progressively more and more dotted looking lines, okay, it's the probability of finding lower and lower levels of Salmonella. And then finally, at 60 MPN per ml, okay, Salmonella, you can start to see now that the level of E. coli does have a rather significant effect on detecting that level. And again, the trend with all those lines is upward. So what does all of this mean? What this means is that E. coli, it's not a great indicator of Salmonella, but generally speaking, as the levels of E. coli go up, the probability of finding higher and higher levels of Salmonella also goes up. And that's kind of a common story with Salmonella generally, which is that if you, indicators are useful, but they're not perfect indicators. And I've got another story that's not part of this, where we showed the same thing with indicators for Salmonella on surfaces in food processing plants. But that's a story for another time. All right, we're going to switch gears once again back to meat. And here we're going to look specifically at Salmonella in beef. And this grew out of a project that I had with a food company that was interested in what would happen if you were to temperature abuse foods for certain amounts of time, what levels of increase of pathogens would you see? And so what we studied here, and we're continuing to do this research to this day, looking at temperature fluctuations and rises and falls over time. So these are predictions for the growth of Salmonella in ground beef under a variety of different warming and cooling scenarios, okay? And so the A panel shows Salmonella in ground beef warming up over 8 hours to different temperatures, right? So starting at refrigeration and then going up to, looks like to about 15, about 25, and about 35, and then cooling down over that same 8-hour period. The middle graph is warming up over 8 hours and then cooling down over 2. And then finally, warming up over 8 and then cooling down, warming up over 8 and cooling down over 4 in the B panel, and then cooling down over 2 hours in the C panel. And what you can see is that the Salmonella really just gets started after about 8 hours, and the cells start to exit lag phase. And then really it's the rate of cooling after that first 8 hours that really does have a significant effect on the risk. And it turns out cooling things down over 8 hours is not nearly as good as cooling things down over 4 hours, and it's even better if you cool things down over 2 hours, and you can really drive you can really reduce those increases. The other thing that's going on in this particular graph is looking at again, in my lab, we study computer models, right? And so one of the other things we wanted to do here was look at the effect of computer modeling. And so what you can see here is that the solid lines represent the corresponding COM-based predictions, and the dashed lines down below are temperature profiles. And so again, what you can see generally speaking is that the models tend to be conservative, right? The models are predicting more growth than you actually see in the food system, although in some cases the agreement is pretty good. So it looks like for the big temperature spikes, the models are over predicting. And then for the smaller temperature spikes, they're pretty much right on the money. And then again, for that lowest temperature spike, the cells basically never get out of lag phase, which is pretty much what the models are telling you as well. So anyway, some interesting work done by Jen McConnell. A little bit about Jen. She left my lab and then went to work in a biosafety level 3 lab studying Mycobacterium tuberculosis. And she did that for a few years. She really wanted to work in the biosafety level 3 lab where you get all dressed up and then go through interlocking doors. And actually, she's now working in New York City Department of Public Health. And I think she's actually doing a lot of COVID-19 work these days because, you know, they're managing a lot of that COVID risk there in New York City. All right, we're going to switch gears and switch topics one more time here. And this is a paper by Tanya. I forgot her first name, Tanya D'Souza. Tanya was a graduate student who worked with me, and she also worked with my colleague Mukund Karve, who's a food engineer in the food science department and at Rutgers, and she studied high pressure processing and peanut butter. We know that Salmonella is very resistant to heat when it's in a matrix like peanut butter. And in fact, what this research proves is that it's also very resistant to an inactivation by high pressure. And so basically what we looked at is Salmonella in unprocessed and then processed peanut butter. And what you can see, the black bars are the unprocessed peanut butter, the gray bars are the processed peanut butter. And then we looked at just plain peanut butter and then peanut butter where we added greater and greater amounts of moisture. And what you can see is that as you increase moisture, so if you can increase the moisture to about 25% and not much happens, after 35% it starts to go down. And when you have peanut butter with 90% added moisture, and believe me, by that point it is mush, it's not peanut butter, nothing resembling peanut butter at all, you can actually begin to see some effect of the high pressure processing. So yeah, so some interesting stuff there, just basically showing that Salmonella is pressure resistant in matrices like peanut butter. Switch gears one more time. We're back to meat. This is a risk assessment in Danish meatballs. Basically, there's a bunch of figures in this paper and tables, but none of them told an interesting story that I wanted to share with you. So instead, I'll talk about the collaboration. This is a collaboration with a bunch of researchers from Denmark. Clady Moller, who's the first author. Clady, she actually, she lives in Denmark now. She's married to a Dane. She actually was born and raised in Brazil. So she speaks Danish, English, and Portuguese. And so she was a graduate student in Paul Dalgaard's lab, or I think maybe in Bjarke's lab. But anyway, she's a graduate student in Denmark, and she came over to my lab to do some research and then do some modeling, and then went back and we published. This is one of the several papers we published together, basically trying to help the Danish. They eat a lot of pork in Denmark, mostly in the form of meatballs. And they've had some Salmonella problems in the past. And so this is some research basically showing that how to basically advising the Danish pork industry on how to best manage this risk. We'll switch gears again back to nuts. This is a paper published by Caitlin Kasuli, Steve Calhoun, and myself. Steve worked for the American Peanut Council. Caitlin's a graduate student at Michigan State University, and I'm actually co-advising her on her PhD. This is some research that we did as a consulting assignment, not part of her PhD. And basically, American Peanut Council wanted to know what the predicted reduction in risk would be for different log reductions for in peanuts, and basically, again, like we did with the almond risk assessment, the chance of greater than one illness per year. And what you can see is that as you increase the log reduction, you basically see a decline in the chance of more than one illness per year from peanuts. On the right-hand side, you see figure 5 there. That's the chance of greater than one Salmonella interest per year as a function of log reduction variability. Right? And so we looked at 3, 4, 5, 6, and 7 log reductions. So starting at the bottom, 3, 4, 5, 6, and 7, just moving up. And basically, we were looking at the effectiveness of that uncertainty. So remember, in that first paper with Daniluk, we assumed that it was a 5 log reduction plus or minus 1 log. Well, what if we could make that process more tightly controlled? What effect would that have on the reduction in illnesses? And it turns out it actually, as you increase the variability of the process, that does increase the risk, because of course the risk is always in the tails, right? So if you increase the variability, you don't really care about the product that's over processed, but you have more and more product that's under processed. So that's going to drive the risk up a little bit there. Here's some paper on sunflower seeds. Now, Salmonella, as far as I know, has not been a problem in sunflower seeds, but this was a collaboration with Dr. Kapali and myself, as well as colleagues at ConAgra. And ConAgra has been very progressive about using modeling and using risk to study safety of different processes. And I didn't know too much about sunflower seeds before this project, but basically what they do with sunflower seeds is they take them and they soak them in salt water, and then they take them through a drying process. It's a somewhat proprietary process where they dry them in this, after saturating them in salt water, they dry them. And so what we were interested in showing was, they roast them. And so what we were interested in showing was what is the log reduction at various roasting processes. And so what you can see in figure 2 on the left-hand side is the 95% prediction intervals for the roasting time to deliver a 4 log versus a 5 log reduction. And the good news is that basically this process is way less than what they're actually using in the industry to roast sunflower seeds. And so this basically was just their validation that their roasting process for sunflower seeds does control Salmonella. Over on the right-hand side on figure 4, you can see this is the geometric mean expected cases per 1 million servings. So basically what this says is that if you're giving it the typical process, you basically are expecting to get about 1 case out of every 1 billion servings of sunflower seeds. So some good news there for the sunflower industry, at least for people that follow a process like ConAgra. I've got a few more slides, and now we're kind of getting into the interesting part, or to me, the more interesting part, because this is stuff that you guys are, you're not seeing it for the first time, but not a lot of people have seen this data. And so this is some data. This is going to be a bunch of different slides collected by my former PhD student, Jin Jung, who actually is now working at UC Davis in Linda Harris's lab as a postdoc. After she finished her PhD with me, Jin did a bunch of research on flour. And this is basically looking at the survival of Salmonella PT42 in whole wheat flour. And basically what you can see is that it survives in flour for a long time. We inoculated about 5 log per gram, and after 25 weeks or 27 weeks, we still were above the detection limit and had about 2 logs, so about a 3 log reduction over that multi-week period. So Salmonella loves to hang out in dry environments, and flour is no exception. Here's some different paper with the same sort of theme. In addition to studying survival in flour, we also were wondering what would the risk be if you made some homemade Play-Doh. And so Jin went out and found a bunch of different recipes for homemade Play-Doh with different levels of salt. And we inoculated these Play-Doh mixtures with Salmonella, and then we studied their survival. And basically the salt content doesn't matter. Salmonella really does not like hanging around in that Play-Doh. We were afraid that it was actually going to grow. Turns out it doesn't really like to grow in Play-Doh, so that's good news. And it definitely does not survive nearly as well in Play-Doh as it does in flour. So you can see we're from 6 logs down to about 3 logs in only 7 weeks, which is much faster than you see in whole wheat flour in this particular experiment. And then the last one, this was interesting. This was actually inspired by a video we saw on the internet of somebody claiming, hey, if you want to make raw cookie dough and you're worried about microorganisms in your flour, you can just simply heat your flour in your oven for 5 minutes and it will take care of all the pathogens. And we thought to ourselves, that doesn't seem quite right, because we know that pathogens like Salmonella do not survive very well, or they do survive very well under dry conditions. And so if you killing Salmonella in a beef patty that you're cooking on your stove is much easier than killing Salmonella in a dry product like flour. And so what we did was we took a bunch of different flour products, we inoculated them with relatively high levels of Salmonella. We used a little toaster oven, a laboratory toaster oven that used to live in my kitchen here, but now resides in my laboratory, and it's for research only. We put Salmonella into it, and this just basically shows a variety of different oven temperatures and heating times out to 7 minutes. So starting, we looked at 300, 350, and 400. And what you can see is that actually it works surprisingly well. And we were able to get a 5-log reduction within about 5 minutes, at least for heating it to 400. You don't quite get a 5-log reduction at 350. At 350, you actually have to go out about 6 minutes. And then even at 7 minutes at 300, you don't quite get there. But it gets you close. And so the good news here is that this video on the internet is not completely without merit. You can actually drive down the concentration significantly in flour heated in an oven. And I should say this is a thin layer of flour, and it's a preheated oven. And again, it's a fascinating bit of research which we're hoping to publish sometime in the next year or so. So with that, more or less right on time, that brings me to my conclusion slide. Does Salmonella offer job security for food microbiologists? I would say yes. You can see a variety of different food products, lots of interesting experiments to be done still. Again, just to highlight some aspects of the work that I do, we like to build computer models, so I talked a lot about that. We like to do quantitative microbial risk assessment, so I talked a little bit about that. And one of the things that we really love doing is collaborating with people, and so hopefully I've highlighted that these are team efforts. Some of it's myself working with a graduate student, but a lot of times it's working with a team of people, even in other countries or around this country. And again, I'll say there's still lots more work to be done. And with that, I'll take it over to my concluding slide and ask if there's any questions. Interestingly, we have not done any work with Salmonella in eggs. I started off wanting to do some research on Salmonella in eggs at the beginning of my career, but my lab tech talked me out of it, and we ended up doing research with Yersinia enterocolitica, which is a psychotrophic pathogen instead, which was a lot of fun as well. So no research with eggs yet, but maybe that's to come. All right, that's me, Laura.
Great. Thank you so much, Dr. Schaffner. I think it's so interesting to see the many food products and non-food products that Salmonella has been associated with. So very well done and informative. At this time, we'd like to take some time to answer your questions. So if you do have questions, please put them in the Q&A box. You'll find that at the bottom of your screen. We'll go through a few of those now. Our first question is for our presenter and our panelists. If you are aware, are we about over on the latest Salmonella outbreak attributed to onions? So Dr. Schaffner, do you have any insights on the Salmonella outbreak?
Yeah, one thing that CDC does sometimes with these outbreaks is they'll put a little, I love looking at those outbreak epi curves, right, where you see the outbreaks, the cases over time. One of the things that CDC will sometimes do is they'll put bars on those curves saying, cases in this window may not have happened yet. Or something a little more fancy sounding than that, but that's the basic idea. And they haven't done this with this particular outbreak. Based on my looking at the curve and thinking about the shelf life of onions, and also knowing a little bit about how Salmonella dies over time, actually, I've done some back of the envelope calculations, and I thought about presenting them today, but I ended up not doing it. But so that's a long wandering way to say the answer is I think the outbreak is over. We may still have some cases. Certainly, I don't think anybody is eating onions that are part of the outbreak anymore. We still may see cases arise that we are learning about just through typing of or sequencing of the strains. But I think that in terms of actually people getting sick from onions, I think we're done with that. The numbers may rise still a little bit though, in my opinion.
Great, thanks for your insight on that. Our next question is, why do you think the USDA is testing for Salmonella in catfish when this fish is usually cooked before its consumption?
That is a good question. I don't know. It may be political. I know the whole catfish issue is incredibly political. With respect to how the industry wants to be regulated, and domestic catfish versus imported catfish. But it's not completely without merit to test raw foods that are going to be cooked. So, for example, we test. We have the Salmonella performance standard for raw meat and poultry, right? So it's not without merit. The idea being that, even if let's say you're going to properly cook your catfish, but you bring it into your home and first you cross-contaminate your kitchen, right? Because there's Salmonella on the catfish. And so there is some benefit to be gained from testing the raw material and driving the levels down. I think in terms of the performance standard, though, and probably this is true for catfish as well, although I am not up to speed on the catfish regulations. What I would really like to see happen with raw meat and poultry, and with catfish as well, is like the Salmonella performance standard is a prevalence. Like, is it there or is it not there? And I would like to see USDA move to a quantitative test to tell us how many Salmonella are there, because there is evidence to suggest that while the prevalence does drive the risk, the concentration also drives the risk. And so I would be much more interested in knowing not just the prevalence of Salmonella, but what fraction have high levels of Salmonella. And then let's focus on getting those out of the food supply, because I think they pose a higher risk. So again, that's just my opinion, but I think it's based on some science and some risk models that I've seen.
Great, thank you. The next question is about the research that you conducted in response to the online video about heating the flour for Play-Doh. Did the flour seem to have the same qualities after heating?
That's a really good question. We did not test that, right? So obviously, to really do a good test, you would need to take some flour that was not inoculated with Salmonella, heat it in the oven, and then use it for baking. Actually, that's a good question. My wife is a big baker. Maybe I could convince her to try that out on the next loaf of bread that she bakes. But it's a really good question, and the short answer is I don't know.
Okay, thank you. Our next question is, can you briefly talk about the impact of cold temperatures on Salmonella? This person's area of concern is specific to ice in a beverage station. Is there any data for survival times in ice?
That's a really good question. I don't know of any data, although I know there has been some research that's been done with ice over the years. I know we've had outbreaks of norovirus linked to ice when it's made with contaminated water. It would not surprise me a bit if it survived really well. Like, what we do to keep all of our cultures alive in our laboratory is we get them really cold. We put them in a minus 80 degrees C freezer, and we put them in something that's going to help them survive some buffer and ethylene glycol. But it wouldn't surprise me a bit. One of the papers I didn't talk about, which was also research that Celia Dominguez did. She's the one that made the growth models in poultry. She also looked at survival on frozen poultry, and it turns out it survives pretty well. You get an initial hit during the freezing process, depending upon how rapidly you freeze. But once it's there for a long time. There's also been documented outbreaks from you saw on the outbreaks list, frozen entrees, frozen chicken entrees that look cooked that are not in fact fully cooked. They get microwaved and not fully reheated. So I don't know of any data, but it would not surprise me a bit to find that it survives really well on ice.
Thank you. We talked about a lot of different food products and non-food products, and the next question is about Salmonella for pet food. So do you have any comments on Salmonella in puppies or dogs and the risk of transmission to young children living with these pets?
Yeah, that's a really good question. I'll just take a brief opportunity to plug my podcast once again. I do a podcast called Risky or Not, and we had an episode recently where we discussed whether puppies were risky or not. It turns out puppies are not risky, but we also I think we did an episode on dried pig's ears as pet treats. We concluded that they were risky. So so pet food is definitely risky. Raw, and there's a lot of interest around raw pet foods. There was a recent outbreak linked to raw pet food, and then some of these dried products, dried pig's ears, beef pizzle, things like that. So I think there's a risk, and obviously if you have puppies in the house and you have children, I would definitely be cautious about that, just like you would if you had pet turtles, right, or backyard chickens and kids. You do want to be very careful about keeping them separate and practicing good hand washing and all of that. So I think there's definitely some risk to kids there.
Great. Let's dive into some different pathogens for a moment. STEC versus Salmonella. Which would outcompete which one? And are you aware of any multi-pathogen outbreaks?
That's a really good question. Let's take the second part first. I'm not aware of multi-pathogen outbreaks, but I am aware of multi-strain outbreaks. So for example, the Jensen Farms Listeria outbreak, they had multiple strains of Listeria. And so it's not common, but it's not unheard of to have multiple strains. I have never heard an example of a multi-species outbreak. Right, that would be an interesting one. And in terms of competition, I don't think STEC and Salmonella behave all that differently from one another. I am really interested in Salmonella versus Listeria, right? So Listeria obviously grows under cold temperature conditions better than Salmonella. Salmonella stops growing at 5.3 degrees C 81F, as I said earlier, but Listeria will grow below that. And then at one point for a project, I calculated where the transition was, and I think it's around 17 degrees C, so a little bit less than room temperature. At that point, the Salmonella starts out competing the Listeria. But in terms of E. coli, I don't think there's much difference. Again, one of the things that I like to do is research on modeling, and people can certainly go to go to combase.cc, the Combase model, and that would allow you to do a side-by-side comparison of E. coli versus Salmonella at whatever temperature you choose, whatever conditions you choose, to see which one will outgrow the other. But my guess is they're pretty close.
Great, thank you. And let's move to talking about Campylobacter versus Salmonella, if we could. Do you have any information on the comparison of prevalence of Campylobacter versus Salmonella? I think this question is specific to foodborne illness and the prevalence of each of these pathogens in there.
Yeah, I think it probably depends upon the food. We know that it's at least based on the most recent, well, based on some historical data, Campylobacter is much more prevalent than Salmonella on poultry, right? So I don't know, haven't looked at recent data, but I think it's going to be food specific. The good news is that well, I guess the bad news is Campylobacter might be more prevalent. The good news is it's kind of a wimpy organism. It's relatively easy to kill via heat. With Campylobacter, I'm much more concerned about if a Campylobacter comes in on the raw chicken, it's pretty unlikely that you're going to undercook it. But given that it may have a high prevalence and the concentration may also be high, I'm a lot more worried about spreading it around your kitchen and getting it onto other things that you're not going to cook, like your salad, or even just getting on your finger or on your refrigerator, and then putting your finger in your mouth later, or something like that. So I'm with Campylobacter, I'm much more worried about cross contamination. But with other foods, I'm not really sure. I'm trying to think of any recent outbreaks with Campylobacter not linked to poultry, and none are coming to mind. But actually, Campylobacter, so I work with a variety of foods, as you saw. I work with a variety of pathogens as well. Campylobacter is not one that I've actually worked with all that much, although I do keep up with the literature. And there's some real good risk assessments out there for Campylobacter in poultry. But other than poultry, I'm just not aware of any data. But there might be data out there, I'm just not aware.
Thank you. And it looks like we've got time for a few more questions. Can you speak any about antibiotic resistance patterns in Salmonella?
That's a really good question. Because I need to focus my efforts, I have not really done much research on antibiotic resistance in Salmonella. I did participate in a conference a number of years ago that was organized by a colleague at Ohio State, and I'm blanking on her name. It's terrible. But so there's a lot of smart people out there that know a lot more about antibiotic resistance in Salmonella than I do. I do know that there's differences of opinion about this. I know subclinical, subtherapeutic levels for feeding for animals is controversial because it may create antibiotic resistance. But again, it's not an area that I've really dabbled. I'm really interested in making Salmonella antibiotic resistant so that we can culture it in the laboratory on non-selected media. That's really a very important part of our experimental designs. That's kind of where my interest in antibiotic resistance begins and ends.
Certainly, great. Well, thank you very much. And it looks like we are winding down here, and we're coming towards the end of our time today. So I know that there are still some questions that we didn't get to answer. I would be happy to pass those along to Dr. Schaffner. you can send me an email at the end. I'll put my email up here in just a moment, but I do want to first sincerely thank Dr. Schaffner for his time and valuable insights on today's webinar. Thank you to all of the attendees for their time and participation and dedication to the food safety profession. What I have on the slide now is information on our next webinar coming up with Eugene Evans from New York State Ag and Markets. He'll be talking about seafood pathogens, so if you're interested in that, please join us next week on Tuesday. And with that, I will say thank you very much. You'll be receiving a survey for comments on the webinar today, and thank you all very much. Be well, stay safe, and we'll see you next time.
Bye, thanks.